Hi, my name is Nur Nadira and I will explain about the protocols. We'll start by preparing electrocompetent bacteria, where we streak DH5 alpha strain onto an agar plate. Then, we inoculate the freshly grown E. coli and incubate them with vigorous shaking. After collecting 5 tubes of bacteria, the cells are pelleted by centrifugation, and the pellet is resuspended in ice-cold sterile water. These steps are repeated 3 times, and for the last washing, the pelleted cells are resuspended in ice-cold distilled water. The next procedure is electroporation. Properly washed plasmids are added to suspension, then inserted into pre-chilled cuvette for electroporation using BioReagent pulser. After the cells have been pulsed, SOC medium are immediately added, then transferred to a tube and incubated with vigorous shaking. After that, suspensions are centrifuged, resuspended, and aliquots of the cells are spread onto LB plates. Then, the grown colonies are calculated. The next procedure is generation of standard curve for plasmid copy number determination. Standard sample qPCR, chromosome standard with plasmid sample is mixed by a fission dilution. Then, another 6 series of dilution is made. For X reactions, 2 different mixes using chromosome primers and plasmid primers are made, then each diluted sample are added to all the qPCR tubes. After that, the reaction was run by the following cycler conditions. Next procedure, we need to lysate standard sample qPCR. A single colony is inoculated into a liquid LB medium and incubated with a shaker and left overnight. The overnight cells is transferred to fresh liquid LB medium and incubated again. Cells then centrifuged. Pellet is resuspended in PBS, then centrifuged again. This step is repeated for 1 time. After PBS is removed, the pellet is left to dry. Then, water is added to the dry pellet, vortexed, and centrifuged. Initial dilution is made by transferring resuspended cell to water, then centrifuge. This step is repeated 1 time. For X reactions, 2 different mixes using chromosome primers and plasmid primers are made. Then, each diluted sample are added to the tubes. After that, the reaction was run by the same cycler conditions.